science courses through which students gain valuable experiences that are necessary for future work and employment. However, COVID-19 pandemic has created great uncertainties, especially in education, where traditional lab-based disciplines, including science, are significantly affected. Therefore, virtual labs cannot entirely replace physical experiments. However, they provide means of performing experiments to run as normal despite the unforeseen circumstance. In today's demonstration, we will provide you a quick overview of the basic science experiments that we at the University of Fiji have been using for our science courses. Uh, First and foremost, I'd like to invite Professor Shaukat Ali, who is a professor in IT and a former dean of SOST, to deliver his welcoming remarks. Over to you, Professor.
Yep. Thank you, Ms. Jafia. Good morning, guests and colleagues. Think about, I have two drivers coming from Suba to Nandi International Airport. One driver is very experienced, driving his car over the 5 years. And second driver just started last year, December. During the travel, both got one big pig on the street. Who can manage this incident properly? Definitely, your answer is, who is driving the car over last 5 years? Without hit the pig, that person can manage his car come to Nandi International Airport. How? Is it magic? No. The things came to him from experience. Because over the 5 years, he drives many times from Suba to Nandi International Airport. He has faced this kind of incidents many times on the street. That's why he managed smartly. This is called learning from scenario. In other words, we call practical learning. How does it happen? It happens in the lab. If we don't know what are the ingredients in water, then how come you expect me to produce hydrogen power from water? We should know what are the ingredients in water, only then I can produce hydrogen power to meet the community demand. So this is science. Learning from laboratory, learning from real life experience, this is science. Science is not logic. Of course it is logic, however, we have to prove into laboratory. So my comment, science is not only logic. If logic, that might be philosophy. We bring the logic to laboratory to prove, then we call it science. So learning into laboratory as a science academic, it is very important to us. You may recall my word, managed driving. So you know, over the world, we are facing the same problem, COVID-19. Still, it is a big problem. As a driver, I need to know an alternative of my learning. So I cannot come to university, I cannot go to lab, even our lecturer couldn't come to give the real life demo to the lab because health is our priority. Now, how we could overcome this crisis? That's why we came today to tell you. I would like to say thank you, Heather and her team. They made a fantastic tool beyond labs. Using that technology, you could have same smell of your laboratory learning. This is important. A scientist should have an alternative. If my kids are scary to take the injection for COVID-19, then they have a tablet. My daughter can take the tablet. This is an alternative. So as a scientist, we need to, offer an alternative. Since we cannot go to lab, they have an alternative. We have to use it. And at the University of Fiji here, we proudly say, out of the 3 universities in Fiji, we are the only one who is using this kind of facility to meet the gap, to help our students, to bring maturity in their learning. So that's our aim. So I would like to welcome in this session everyone who are listening to us. After this session, who would listen to our recording as well, I would like to welcome you to stay with us and learn how are we helping to our students during this very difficult time. On behalf of the School of Science and Technology from the University of Fiji, again, I welcome you. Please stay with us and learn how this wonderful technology is working in the virtual domain. Thank you very much. Over to you, MC Jafia.
Thank you so much, Professor Shaukat Ali, for your uh, welcoming remarks. Okay, participants. So next up, we have Heather Miller from Beyond Labs. She is a physics instructor and an instructional designer, and she will be providing us a quick overview of this Beyond Lab platform that we have been using at the University of Fiji. So over to you, Heather. Thank you.
Thank you so much for that introduction. Let me introduce myself. So I live in Boston, and it's freezing here. I'm admiring all of you looking warm there. I'm so jealous of your amazing weather there. I helped design these labs when I was a physics and chemistry student decades ago at the university, and I was taking all of my science labs just like normal labs used to be given then. And I was working with a professor who was the major creator of the labs, Dr. Brian Woodfield. And his basic concept of why we decided to develop, he asked me to then work for him, and I've been working for him for the last 2 decades. His major concept in developing these was to give students a chance to practice in a safe, easy environment where they can make mistakes and learn by making mistakes. Because students are usually scared to make mistakes, and we as their instructors are scared when they break our equipment or they completely don't do things right. And so the initial concept in developing these was not to replace labs. We, of course, did not picture the pandemic happening back then, but we developed them 2 decades ago because we wanted to create a practice environment where students could have fun and they could explore and ask questions and try things that in a normal physical laboratory you couldn't do because you don't have enough money and you don't have time and you don't have that flexibility to let students really explore and make mistakes. So that's what it was designed for. It was a simulation just to give students the practice, like your story about practicing driving, so then they're ready when they do get to go back to a real lab. And oh, we hope they get back into the real lab soon. There is nothing like being in the real lab. There is no way we want to be a lab replacement. That's not our goal. We want to be a lab practice. I was just talking to a professor this morning who said he's having his students practice every day in our labs, and they have the luxury where they are to be able to go into the lab. And he said their instructors are seeing that the students that practice in our labs are doing so much better in the real lab because they've learned the type of skills they're going to be doing in the real lab, and then they're ready. And they go in and they can have a real lab experience, and they're confident because they know what type of data they need to take, and they can get in and do the real science that we actually want them to do. So I do hope you hear that message out of what I'm saying of let's get them back in the real lab as soon as we can. But this is a useful tour or useful tool on the way to be able to prep them, to have them practice. And then I hope you'll also see how it's useful for homework and it's useful for exploration in ways that you can't do. The principle of the virtual labs is that they are completely open-ended, which required some pretty crazy programming on our end. So it's not what we call a lab on rails. Students can go in and they can mix any chemical they want. And they can go in and they can completely make almost any mistake we could possibly program in. And then they have to, by analyzing the data, just like a real scientist would, figure out what they did wrong based on their results. So our simulations are what we call open-ended because we don't have them saying, do this next, do this next. It's not like a click next simulation. It is actually a lab where they can move glassware around and choose this and weigh this and try lots of different things. So for those of you who have seen various types of simulation, this is a lab where they can go in and do anything. And we're not going to tell them what to do unless you as the instructor want to give them step-by-step instructions. So that is your privilege to decide how much hand-holding you want to give to your students in our labs. So the basic concept of how we have them structured is we have our labs. We have lots of different lab benches, which are like little simulations. So we have like a calorimetry lab bench and an inorganic chemistry lab bench. And my favorite, the physics mechanics lab bench, where you get to do ramp experiments and ball experiments and a lot of biology different experiments. And they can go into these different simulations, do whatever they want to do, collect data, and then you as the instructors get to decide what you want them to do with their data. Whether that is in collecting, filling out, we have some standard worksheets you can use if you want to, but you don't have to use our content. Most instructors in the US and in Europe and in South America write their own worksheets because teachers like to exactly decide what they want their students to learn. So you can have your students go in the lab, you tell them what you want them to do, they collect their data, and then report it back to you just with a normal lab report, just like what you would normally do. So that's the basic structure of what we're trying to do. Open-ended lab, they can go in, do whatever you want them to do in the lab, collect the data, send it back to you. I know you're going to have other professors showing how the labs work. I didn't know if you wanted me to like actually show anything in the lab or just talk about the basic principles. So feel free to stop me, say, Heather, we want to see this, whatever. I'm happy to show you whatever you want, but I wanted to give you an intro into the principles of the lab and what we're trying to do here. So please tell me what you want next.
Thank you, Heather. I think that was well explained. I'd like to reiterate that most of the times you don't have enough time for all of the students to actually carry out the experiments or the resources. So these virtual platforms give students their own time and space to practice on their own. So thank you for summarizing that. And I think that is well understood by all of us. And thank you so much. Yeah.
You're welcome.
So moving on, we would just like to capture this moment. Will you be able to take a photo of us? I'll stop sharing my screen so that we have the participants and the speakers. Can we have you take a photo of us so that we can capture this moment for future reference. Participants, kindly requesting if you can turn on your camera. If you can't, then it's okay. So whenever you're ready, Krishna will just give us a heads up and we'll smile. Demonstrations will include biology experiments, chemistry, physics, and environmental science experiments. So we are just showcasing some basic experiments to show you how this Beyond Labs platform is and how we are using this for our courses. So I'll hand over the platform to our biology experts, Ms. Priyatma Singh and Mr. Danian for their demonstration. And just like Heather said, if you have any questions regarding Beyond's Lab or anything in between, you can use the chat box. You can always type in your queries, your questions, and we will be able to respond to you either through the mic or through the chat box. Over to you, Priya.
Hi, Safia. Are you able to hear me?
Yes, Priya, loud and clear.
And are you able to see the presentation, my slide?
Yes,
Sorry, my desktop.
I can see the screen, the Beyond Lab screen that you're sharing.
Okay. Okay, great. So a very good morning to everyone. It's indeed a pleasure to be here and doing this demonstration for our prospective students so that they can understand what are the virtual lab platforms that we are using at the University of Fiji. Just to let you know, we also started using the Beyond Labs platform just last year. And we before that, as Professor Ali mentioned, we have been doing face-to-face practicals. But then realizing that we need to give our students the experience that they need, the laboratory experience they need, we reached out to the virtual lab platforms that are available out there. And then we subscribed to Beyond Labs, and we have been using Beyond Labs platform since last year. And we, as instructors, find it extremely effective platform, online platform for demonstrating the labs that we were used to doing during our face-to-face sessions. So I'll basically give you a glimpse of the labs that we actually do in biology. Me and my colleague will try to show you some of the simulations today. Of course, there are a number of practicals that we can do in the biology lab platform, this virtual lab platform, but we won't be able to show you all of them today. So we're just going to show you some glimpses of the labs that we do in biology through this Beyond Labs platform. So as you can see, this is the main platform, and we have two features here, web-based option, and then the other one is the desktop option. So we can access labs through the web, but we can also access our labs through the desktop. So for biology, we have this option where we have to install a feature that allows us to access our labs via our desktop. So if you click on it, it takes you to virtual biology, and I hope all of you can see this screen which says welcome to virtual biology. So there are a number of opportunities here available to all of us in biology: microscopy, genetics, molecular biology, ecology, and systematics. Um, in these different biological labs that we have, there are a number of experiments and practicals that we can conduct. So today, for instance, I'll just very briefly show you what we can do when we access microscopy labs, or what we can do when we access genetics lab. We also have this option where we can do molecular biology labs, and there are slightly advanced labs which our students who are in year 2 or year 3 are able to access and conduct. We also have ecology labs, and this will be demonstrated by my colleague Daniel later today. And I will also be very briefly showing you what we can do if we access systematics lab. So I hope you're all with me. So I'll take you through the Microscopy Lab biology virtual lab, and it will take you to a lab, a virtual lab that has all these different microscopes. Okay, um, so you know, in our labs here in Fiji, in most of the universities, we can only see three labs. This three microscopes, which are the Fieldscope, of course, the Dissecting Microscope, and we have compound microscopes in our lab here in Fiji. But in this virtual lab platform, you can see that we also have scanning electron microscope and we also have transmission electron microscope. So these are actually very expensive piece of equipment that we normally do not have here in Fiji. But with the help of this platform, we can actually observe specimens using this high magnifying microscopes that we have here on this platform. So we have a species selector here, and these are the different species that we can select for observation. You can see there are a number of species to select from. They belong to plants group or animals group, fungi group, protista, bacteria, archaea. Now, some of these specimens we don't even find during our, lab face-to-face laboratory classes, but they are all here. So let's say you want to observe a particular specimen. So your instructor gives you a manual or a instruction sheet. So normally at the University of Fiji, and as Heather said, we instructors love to design our own instruction sheet, and we give it out to our students in advance so that they can go through it. They can do the experiment on their own. They're going to make observations. For instance, in biology, we give them a number of specimens to observe, and they can do so on their own before we have the actual virtual lab demonstration. So it kind of gives our students ample time and space to make these observations or do these experiments on their own. So when we have actual experimental dates, then they come prepared for the class, and they know exactly what they're supposed to do. So if there were, for instance, some problems that they were facing when they were trying out these experiments on their own, they can, clarify all these queries quite quickly. And, the laboratory session then becomes very easy to conduct, both for the instructor and for the students. So I'll just give you a brief demo here. Maybe let's pick one specimen, and the one that we all are perhaps very familiar with, and it's a very favorite lab specimen is onion. So if you have selected onion, you can see that the the specimen has been picked up by the Fieldscope. Okay, it's been picked up by the dissecting microscope, and it has been picked up by the compound microscope. Now, if we were in a face-to-face laboratory class, you would be, using these prepared slides, and you would be, focusing it under the microscope on your own, and then you'll be observing basically the same thing that you are observing here. So these diagrams that you see here, these photos, pictures that you see here, are basically the same thing that you would have observed in a real laboratory class. Okay, so Fieldscope, of course, before I go into the details of this, just a brief introduction to the different microscopes that we have, and this clearly shows you the resolution power of these microscopes. So these are the additional information that is provided in all the laboratories, biology laboratories, in fact. And these are like those little, add-on information that helps you to understand your practicals even better. So let's say we are observing, of course, the Fieldscope will, it's something that you can use to observe with unaided eye. And of course, this shows you what you will see using a Fieldscope. So the different, you have so many options here, so many pictures of the onion that shows in detail. Sorry, that's a garlic. So these are what you see. These are diagrams that you see when you use a field scope. Okay. Now, say you want to see this same onion under the dissecting microscope. So we switch that off, we switch on the dissecting microscope, and it allows you to observe what you would be observing if you dissect the onion and try to observe it under the dissecting or stereoscope microscope in the real laboratory. So there are different angles to the specimen that allows you to basically have a good look at the dissected onion under the dissecting microscope. And it also shows you the magnification, and there are labels as well for some of the diagrams and descriptions that helps you to understand what it is that you are observing. Then let's say, okay, I have been asked by my instructor to observe something under, let's say, compound microscope, and this is what we normally have in our labs, and we normally ask our students to observe things under a compound microscope. So it will show you what you are observing inside the onion cells. So these are the various onion cells, and in fact, you can clearly see the different stages of mitosis happening within the onion cell. So it shows you what the cells that are undergoing the cell division process looks like under the compound microscope. It gives you the dimension, it gives you the magnification, and it very neatly shows you. So for instance, this cell here is undergoing metaphase stage, so you have some information about metaphase. Now you see this stage in the onion cell, and you can see, okay, so this is how it appears under the microscope, under the compound microscope, that is clearly anaphase. Sometimes we remove the labels, and then we ask our students to identify the different stages on their own. And there are various diagrams that shows the different parts of the cells, quite a detailed picture, I would say. Sometimes it's not even this clear in the laboratory, but here we can clearly see the cells, we can clearly see the different stages, etc. And students can, of course, save this. So for instance, this is the diagram that is required. The instruction sheet requires you to draw this, label it, understand or describe it. So you can always save it, and it gets saved in a lab book that we have here. So we have a lab book, and the lab book will save your results that you can access later as well. So this is basically, what we can see or observe using the different microscopes. Of course, we can observe specimens under electron microscope, or scanning and transmission electron microscopes as well. So, for instance, I was trying to see, which one allows you to observe detailed picture under the scanning electron microscope, and I wanted to observe, for instance, let's say in your instruction sheet, you have been asked to observe an earthworm. Okay, so the field scope has picked up the specimen, and the scanning electron microscope is showing a detailed picture of the earthworm as well. So you can claim that this is the resolution or magnification that the others in the laboratory, the other microscopes in the laboratory, will not allow you to see. So this is here, you can see satay here, and this is basically quite magnified under the scanning electron microscope. So you can see detailed diagram. These are the bristle-like setae that they use for crawling. Then you can see the level of detail that's shown here. So this are some of the labs that you can see using the different microscopes, or specimens that you can see using the different microscopes in the biology microscopic lab. And this, if you want to exit this, if you've conducted the experiment, you can exit it, and it will take you back to the main virtual lab platform. You can observe labs, genetics lab as well. So there are experiments that we do in genetics. Or For instance, you might be familiar with the Mendelian genetics or population genetics. And it's very easy to conduct these kind of experiments using the genetics lab platform that we have here. So, we I just wanted to also show you that we have sometimes preset labs as well that we can do, but here you can pick whichever specimens you want and you can make genetic crosses. You will be able to get results that will be very easy to interpret. So for instance, a pea plant, I'm sure all of you know about pea plant and the 7 characteristics that we often study in the Mendelian genetics. So let's say you want to study the characteristic, which is flower color. The instruction sheet will have the different genotypes for the parent 1 and parent 2. You will be asked to fit those alleles here in this Punnett square, this virtual Punnett square. The instruction sheet will perhaps ask you, that you have to do this. You have to come up with 100 offsprings, and you will have to understand the results as explained by the 100 offsprings in the F1 generation. So you do the cross and you get your results here. You can save these results. So for instance, what we do is we normally give these different types of genetic experiments in the instruction sheet, and we ask students to first attempt these on their own, and then they can cross-check their results with these monohybrid and dihybrid experiments that they do virtually. And these are simple examples that I'm using here, but you can also do complex patterns of inheritance exercises as well. So there are a lot of complex genetic crosses that you can do, and it will help you to understand the results even better. Okay, and like I said, molecular biology has advanced genetics lab, and this is the virtual lab platform that we use to conduct our DNA analysis experiments, to do our polymerase chain reaction experiments, and our year 2 and year 3 students are more comfortable using this platform because they have the knowledge and the ability to conduct advanced molecular biology experiments. Ecology will be explained by my colleague Daniel after me, but I can very quickly show you the experiments that we do in systematics. So systematics is a branch of biology that deals with taxonomy and that deals with the classification system of the animals and plants and the other various species that we have. And students normally find it a very tedious task, but this virtual platform allows us to kind of narrow down the different classification systems that we have, and it helps you to understand the taxa or the groups for each species. So for instance, you want to find out about, let's say, if you move on to animals, you want to find out, you want to identify or classify a particular organism or animal, then you can select it, and you can do an experiment on your own. So for instance, that cat belongs to a eukarya domain, right? And it takes you to domain eukarya. You can see it lists what are the different supergroups that are under domain eukarya. That, once you know the domain, so this is something that you need to know on your own, that you would be able to, for instance, identify these different species using these different classification systems on your own. So it helps you to understand what are the groupings for different animals. So for instance, for cats, we move on to the Euconter group, and we move on to Opisthoconter, and then it takes you to the next stage. It's a metazoa, and then it's a new metazoa. So it keeps on classifying the species, and it can take you to the very last level where you can identify the genus and species name of the specimen that you're observing. So it's a very useful tool which sort of helps you to make your own classification diagram for different species quite easily, conducting using this virtual lab platform. You can save them and you can access them later, or you can print them and learn them as well. Okay, so these are some of the practicals that we can conduct using the virtual lab platform for biology labs. I think I will stop here and give the opportunity to my colleague Daniel to explain more about some labs that we can conduct using the Ecology platform. Thank you, everyone.
Good morning, all. I hope everybody is well and safe. Just give me a moment. I'll just share my screen. So the biology experiment that I will be demonstrating is the Ecology Lab Bench. Now, ecology, as we know, is probably one of the most complex levels of biology that we have, right? Because we are studying the mechanics of the environment. And here, it is very difficult to study the mechanics of the environment during a COVID pandemic because we cannot go out into the field and observe organisms. So the Ecology Lab Bench provides you with a picture of how the environment functions and how organisms are related to each other and how they're able to maintain ecological balance. So I'll demonstrate to you one particular type of ecological relationship, that is the predator-prey relationship. Now, remember that this type of observing this type of relationship in the environment takes years of observation and data collection, but it only takes minutes to actually run it in a virtual simulation, such as the Beyond Labs Ecology Lab Bench. Now, as I mentioned, we'll be looking at the predator-prey relationship. So when I say predator, right, the first thing that may come to your mind is a very large, ferocious organism with very large teeth that can kill you instantly. But the definition of predator, we must remember, is very broad. Predator is any organism that consumes another organism, whether fully or partially. So based on that definition, humans too are regarded as predators. And so are mosquitoes, for example. So based on that definition, we're actually going to run a very simple simulation and generate some graphs, right? In the Ecology Lab Bench, one thing that is of an advantage is that the graphs are generated for you, and you only have to interpret those graphs. And interpretation of graphs is very key, right? So usually we have graphs in physics and mathematics where you have to interpret, but in biology too, remember, graphs play a very, very critical role, especially if we're dealing with concepts such as ecology. So the three organisms that we will look at, right, very, very simple, and they form a very simple food chain as well. Now, as year 13 students, you already know that food chain starts off with a producer. And in this case, you have a grass species that is a producer. Then the second organism that falls straight after a producer is a herbivore or herbivory organism. In this case, it's a elk. A elk is a very large, one of the large herbivore organisms that exist in many parts of the world, but it does not exist in Pacific Island nations, right? In Pacific Island nations, we do not have any extremely large organisms. And then lastly and finally is the wolf species. And in this case, the wolf will act as a predator, the major predator. So we will run two simulations and see how the population of the organisms fluctuate. Like I said, this experiment is very difficult to do in a field, mainly because of the COVID pandemic, and you have to spend years and years observing this type of ecological relationship. But using this particular platform, and I mentioned before that we can do it within minutes. So the first scenario that we will try to demonstrate or try to run is without the main predator, that is without the wolf species, and we will see what actually happens to the two populations. So I'll click on wolf and I will remove it from the scenario. So we only have two organisms now, that is the Kentucky bluegrass, which is a very common grass species. It's a lawn grass. And one of the major advantages of this species is that it's grown world over. The major advantage is that it's able to stop soil erosion. And the elk species, which feeds on the bluegrass species. So we'll run the simulation. You can click on the green button here, and you will see that the graph is being generated for you. Now, as the graph is being generated, you can see the population is fluctuating. You will see that the population increases. You will see that the population decreases as years go past. Now, at the moment, the simulation is occurring at a very slow pace so that you're able to follow up with the graph very easily. But if you want, the Ecology Lab Bench also has a feature to accelerate the process. You can click on the plus button here and you can accelerate the process. Now, as I mentioned, interpretation of graphs is very important. Even at university level, interpretation of graphs is key. You can see how the graphs are forming. You can see that there are fluctuations in the population, but you can see that there is a trend that is occurring. Now, if you look at the elk population, the green graph, the green line that you see here, you can see that the population is increasing. Now, you may say, you may ask, why is the population increasing so rapidly? That is mainly because there is no predator controlling the population. Therefore, there's nothing limiting the population. Therefore, the population is increasing dramatically. But you can see, we'll stop the simulation at 29 years, right? So this simulation is it has overgone 29 years. And you can see that the population of the bluegrass species has decreased. It's very difficult to observe this in a natural environment because you have to constantly observe it. And that too almost on a weekly basis to see the significant changes that have occurred. But in this simulation, it's very, very easy. You can basically see that the population has declined. Why has the population declined? Because the elk species are continuously feeding upon the bluegrass species. And thus the reproduction of the bluegrass is relatively slow, but the consumption is high. And this will continue to occur, right? We will continue with the simulation and we will accelerate it. Okay, you can click on the plus button to accelerate the process, and we will try to run the simulation to a point where the population of the two collapses. That means it reaches a point of extinction. Right? You can see that the population of the bluegrass species has reached to a point where it is zero. And we will stop the simulation at 42 years. So 42 years has passed, right? And you can see that the population has collapsed. And so has the elk population because they have nothing to feed on. So remember that in the first scenario, it is important to note that why ecological relationships are so important. And if we remove an organism from the scenario, what will happen to the other populations? And I'm repeating myself here again, this is very difficult to observe in a natural environment. But like I said, it takes seconds or even minutes to observe this type of relationship using the Beyond Labs Ecology Lab Bench. Now, here the population has collapsed, so the elk species has entered into the extinction stage, where it's no longer existing. So that's the first scenario. So we will reset this, and we will introduce, right? You can click on the preset section of the Ecology Lab Bench, and you have experiments that are already present there for you, and you only have to click and run the experiment. Now, you can either use this, or your instructor will give you a certain or a very specific lab handout or instruction sheet, and you have to select the organisms based on those instruction sheets. So we will go back to the first situation where you have the Kentucky bluegrass species, the elk, and the wolf population. We will run the second simulation where the three organisms exist and where you have the main predator existing as well. So you can see based on the graph, the three graphs here, you can see the population of the three organisms are relatively stable. Why is it stable? Because the wolf population is controlling the elk population, therefore controlling the bluegrass population as well. We will accelerate the process. You can press the green button here to run the simulation and the plus sign here to accelerate the process. So you can see that the Kentucky bluegrass population is relatively stable because the elk population has declined to a very stable point, because of the wolf. Now, one of the questions that I usually ask my students is that what benefit does the elk population has? It is constantly being predated upon by the wolf. Now, the answers that I get are mostly correct from my students, but the answer here, the most important point to carry forward here, is that the wolf population is removing the vulnerable organisms from the elk population, right? When we see predations, right? You must have seen documentaries on National Geographic and various other documentaries on YouTube. Predators, what they do is that they usually target individuals that are vulnerable, that are sick, or that are diseased. And by removing these vulnerable individuals, what usually happens is that they are making the elk population stronger and more adaptable. So the concept of evolution comes into the picture here, just by looking at the three graphs, just by interpreting these three graphs. And you can see that the elk population and the wolf population are directly proportional as well, right? The predation will increase if the population number of elk is high. If the population of elk is low, predation will decrease. So interpretation of these three things is very, very important. So we have run the simulation for 49 years. And doing this experiment in the field for 49 years would be basically impossible to do. You'd have to observe, make observations, like I said, every week. But using Beyond Lab simulation here, such as the Ecology Lab Bench, we're able to conduct these very simple experiments within minutes. Okay, and students can actually not just follow the instructor's instructions, they can actually do some of these virtual experiments and learn about these ecological relationships at home as well. Students have access to these Beyond Labs database, these labs at home, using their own laptops or gadgets. Now, another very important thing that I'd like to mention is the control of abiotic variables. As year 13 students, you already know what abiotic variables are. These are non-living factors, such as wind, temperature, pH, toxicity level. So at this point, looking at abiotic variables is not that important, but the information from the graphs is critical. And creating these graphs from scratch for 49 years would take a very long time to happen. So what students usually do is that they save these graphs in their virtual lab book. There's a virtual lab book available within Beyond Labs. And then they magnify or zoom into these graphs, and they make the observations every 10 years, every 15 years, or every 20 years. And they look at how the population has significantly changed over time. So remember that this simulation is not something that we have created or that Beyond Labs has basically created. Remember that this is based on a real life situation, but this provides you with a window as to how these simulations actually work and how will the populations differ over time as they increase or decrease or as years go past. So thank you so much. I hope you were able to really grasp the information that I've tried to demonstrate through this Ecology Lab simulation. Thank you so much. Over to the MC. If you have any questions, please feel free to type it out in the chat box, and the biology team will be more than happy to answer your questions. Thank you so much.
Yay. Thank you, Daniel and Priya, for the thorough demonstration of the biology experiments using Beyond Labs. So if you guys have any questions, please feel free to type in your queries in the chat box and we'll get back to you. I have a trivia question that I'll post in the chat box that you can go through while the demonstration is being set up by our next team, which is the physics team.